Now we're going to do a bit of a practical kind of session of do's and don'ts of TurboBrainVoyager. So this will be Amir and Ethan taking you through. Now I think we'll call this interactive in terms of there are right and wrong answers and there are caramello koalas. So if anyone's really getting hungry, good time to hang around. You might get a chocolate piffed at you. You have to answer a question to get that Sampras.
Yeah, so I think this is a good way to show as well, like a lot of what's gone before has shown how complicated neurofeedback can be and how many systems are in place and how many places it can go wrong. So this is really a collection of myself and Amir's best hits in terms of mistakes that we've made. We thought we've a participant in and everything's going well and then we do something stupid. So yeah, I think Amir, if you want to start the first one.
Okay, so yes, there are going to be a couple of videos, like some of them that we showed earlier, and probably you can guess what went wrong. We can start from this one. So this shows that a neurofeedback experiment has already been started, but do you think that everything is going right in this demonstration?
You can shout out if you think you know what's going wrong. And I'm going to throw a caramel koala at you. Yeah, so the hint is the two boxes on the, is there a pointer? Yeah, there. So these two boxes are empty.
Yeah, so these boxes actually.
What should that be showing? And then that'll tell you what we're missing. This as well will show from Amir's presentation. There should be something here, but there's not. What have we forgot to do here? Close. I'll give it to you. Got it. Yeah, so Amir, do you want to?
Yeah, so as you remember, we had kind of these ROIs that we needed to preload to get the data from, but we just forgot to preload any ROIs and we just started the neurofeedback experiment without any outputs. And now that we want to kind of build the mask, we don't see any mask here because we didn't kind of preload it. That was the answer.
Yeah, so again, looks pretty stupid when you see it up there, but it happens. Whenever you have 20 different things to do, usually something slips. But yeah, I think what we've learned is just to recognize the mistake that's been made quickly. I think if we go to the next one, it should show. Yeah, so this is a functional localize and run. So again, the issue should be first. So if you get the issue right, you get a koala thrown at you.
Yeah, so this is supposed to be that key reactivity task. The key reactivity that we already defined our region of interest, and we wanted to extract those couple of voxels for each individual. Yeah. So yes, we see that this is a kind of normal key reactivity task, and now that's the time that we want to define those specific voxels of interest. Also, do you think that something might be wrong here?
This one's a bit tricky. You probably would have had to be paying really good attention to when Amir showed the proper one in his presentation. But can anybody hazard a guess to what hasn't been done here? So the key reactivity has run. You can see there's an ROI or VOI that we've loaded. And we're trying to convert from VOI to ROI to use the functional localization. This is tough. Anyone, any guess? A hint is it's with the volume of interest box. So there's something that we haven't done inside that box. Do you want to maybe? Yeah.
Something around here.
Okay, maybe we can just show the next. Basically, you have to actually select the VOI. So you need to actually physically. Yeah, so this just show the proper one. So you need to actually highlight your region of interest and then follow these strict sort of steps. So VOI to ROI, and then you click func mask and threshold, and you reconvert then VOI to ROI and save as a .ROI file. So nobody gets a koala for that one. I might have it myself.
So this is the one that you actually found those MNIs from found the coordinates from the localizer on, and now you want to copy them. Probably, maybe I didn't mention that in the presentation, to be honest, that this detailed. So I won't expect anyone to kind of answer, but yeah. So the idea was to kind of copy those coordinates of those voxels into another separate text file, correct?
Can we pause it?
Yeah, maybe. Let's just go back. Yeah, the idea was to kind of copy those MNIs, but maybe just take another look and see if there might be an issue.
Just to clarify, the reason why we need to convert and put it into another text file is because of the confound region. So whenever you convert from your VOI to ROI and you save your .ROI file, that generally will only give you one region of interest, but we want to use two. We want to use our target ROI and a confound ROI that we've already previously mentioned. So what we do, we store it in a .voi file, which is a volume of interest file in TBV. And you copy your functionally localized ROI coordinates to that like already entered confound MNI coordinates. So you've got your two regions. But you can see here that something has went wrong. The ROI is sitting way off the brain, which it's not supposed to. Our ROI is not outside the brain, funny enough.
Yeah, and you see that it results in like crashing TBV, which means that it couldn't actually find the thing. So yeah, can you guess what went wrong?
This also took us weeks to find, so if you get it within a 2-hour presentation, I'll give you the whole bag of koalas. It's just a good example, though, of how you've got so far in your experiment, and something so small can actually crash TBV. It's to do with the temp. Yeah, you know what? Good catch. So it is to do with the template. You can see down the bottom, if this plays again, maybe. But basically what we're supposed to do is copy just until the final MNI coordinate and paste that into the VOI. But what was done here and what we were doing beforehand was copying everything. But there's a little line at the very bottom that says max PSC equals 1.5 or whatever it's been, there. So whatever that's been set as, transferring that crashes TBV. So something as small as that is going to mess up your whole experiment. And we didn't really know what was going on for a while. But yes, it is the template. It is the template file. So this again, you would have had to been paying attention to Amir's presentation, but it's to do with the DICOMs. The hint is it's the only thing that changes in terms of how we label our DICOMs per each run.
Yeah, so what happened was that we changed the DICOM series number from, for example, 30 to 32. And let's say after that timeout second, TBV just said that, okay, I couldn't find any DICOMs coming through. So What do you think was the issue here? Wrong. You started with a good word, wrong, but yeah, should be good enough. Yeah. So we started with the problem was the wrong DICOM series number actually.
Yeah.
Yeah, that series number that TBV was expecting to receive data from, that series number was not correct. So it should be another series number. So TBV just didn't receive anything with that specific series number and said that, okay, I couldn't find it.
Yeah, this is another nuance with TBV is this comes into relevance for all of your runs in neurofeedback, but including your anatomical run. So usually we're in communication with the radiographer, who's usually Beck. We ask Beck, what is the uniden? So we're looking for the uniden to reconstruct our anatomical image at the end of the structural so that we can place our ROI on that. But you have to get the uniden series number. So we ask Beck or whoever the radiographer is, what was the series number? And then that's the series number that we input into TBV. Once you start the neurofeedback, then every run is going to be 2 DICOMs after for us. So if the previous run, you check your target folder, if the last DICOM series number was 30, you have to go into TBV and set up that the expected one is 2 plus that, so it's going to be 32.
So here we are just setting the TBV for starting the run, and now we are receiving this kind of issue, which I hope is not that clear, so you can't guess the error.
The title gives it away.
Yeah, I can go back and start it again. So this was our setup. If you see, do you think everything in the setup is correct? Sorry? Exactly. Yes. Correct.
You're going to have loads of caramel.
So that was the thing. Like we said that we should ideally have a protocol file which has all the onsets and offsets of the condition inside, and we need to kind of load it here, the protocol file. Otherwise, TBV doesn't know what's happening during this volumes, and yeah, it just crashed.
Yeah, so again, it's something, your protocol file, how could you forget to ever put that in there? Like, how stupid can you be? But we've been that stupid. Yeah, so this one is about folders and how many folders have to be correct and right before TBV can actually run.
Yeah, so you see that we actually it's happening at the same time. Like we started TurboBrainVoyager, and at the same time we are starting the MATLAB task. So they ideally should start at the same time, and MATLAB looks for receiving data from TBV to show the bar to the participant. But we've faced this hard moment so many times, to be honest. This is so hard and painful even now. Boom, crashed. What do you think was the issue?
Close-ish. Wrong folder is correct.
Yes.
It's a terrible throw. Don't eat that, Sampras. Yeah, so the wrong folder. So you have to set your target and your feedback folder to the correct location. One of the things also that can happen is we find with our MATLAB code, if it would crash or if the button box, something would happen with the button box, and we didn't set up the run correctly, MATLAB would write an RTP into your, say, RTP1 or run1 RTP folder, which means when we go to set up the run again and we try to run it, it's going to crash because there's already data there. So one of the things that you can check, if you are worried, I wouldn't recommend it all the time because you're trying to get participants in and out as quick as you can, but those folders need to be clear at the start of every run, so they have to be empty. But yeah, this was about just wrong selection. Yeah, this isn't a video, but can anybody guess? First of all, is this gonna run? Will this neurofeedback? Do we think it runs or do we think it crashes? There is a hint in the picture. Okay, the hint is it does run. So you can see on the right-hand side, we are still getting TBV that's running. You can see the time course of these two ROIs with like the red bar. Does this point? No. Does it? It's got a picture of a laser here. So you can see here that it does actually run, but there's obviously an issue because the title is called Guess the Issue. So the issue is not with TBV running per se, but there is still an issue. Can anyone see what that is? Another hint, it's somewhere in here.
And another hint is that when you run the task, that's the time that your script actually crashes and can't show the bar to the participant because it couldn't find something.
It goes back, what are we actually presenting back to participants?
Yeah.
Close.
Like, what was the? Yeah.
So the thermometer won't be displayed, but there's a reason why it won't be displayed is because MATLAB is not able to get the feedback value scores. So what box has not been checked here? It's probably hard to see. Okay, but we'll give you a koala for that. Okay. But it's the log RTPs. Is that RTP? I can't even see.
Yeah, that one.
Whichever. I'm not allowed to throw koalas anymore. It's ridiculous. Yeah, but it was the check RTP. So again, this is something that you've your whole neurofeedback run up and running. Everything is in communication with each other. Everything is correct. You've localized your ROI. You've correctly selected your two VOIs. The DICOM series number is correct. Your target folders are correct. Your feedback folder is correct. But you forgot to check a box. And you're not getting any feedback value scores. So yeah. Yeah, so the last one is more just to go back and to reiterate the point that what we've showed is what can go wrong at the front end, but worryingly, there's a lot that can go wrong with the back end as well. So it's very important that everything, first of all, is in communication with each other. That's one of the key things. Like, we have found the front end, we have everything set up. We think we're happy days, everything's perfect. And then we figure out that we forgot to make sure that the STEM PC is in connection with the MATLAB PC or the scanner. So it's really important to dot all your T's, dot all your I's, cross all your T's. Dot all your I's, cross all your T's at every different stage of the protocol. So at the front end, there's a number of things that can go wrong, but also at the back end as well.
Yeah, and as Ethan mentioned, from some stage you feel that any like feedback calculation, everything is correct, everything is there, like you suppose that it should ideally run. But I would say there are just a couple of things. The one thing that could help us at some stage was just to have a checklist of those couple of actions you need to do before running the experiment, like on the day and before running the task.
To read the checklist.
Read the checklist, go through the checklist, check RTPs, like the checklist that I showed earlier in the presentation. Check RTPs, I don't know, set the right folder, preload your ROI, select your VOIs, just follow the steps for VOI to ROI mask. These are just those sort of like steps that are necessary, and they are big enough to crash your course and result in easily, I can say, like 1 more hour and participant is in the scanner, and it's getting frustrating for everyone and everything. So just keep those smaller steps but crucial ones in mind, and I think neurofeedback will be easier, hopefully.
And yeah, that's it. If anyone has any sort of questions or wants to follow up, myself and Amir's LinkedIn was there, but feel free to have a chat after or reach out to us an email, whatever suits.
Thanks so much.